Hi, Bill Mobley here again, looking at my program for the 2012 meeting of the American Neurological Association to be held in Boston from the 7th through the 9th of October. Let's talk about the 7th. Let's talk about Sunday and what's going on Sunday. After the development course, the career development course in the morning, we'll have two symposia separated by an interactive lunch workshop. The first symposium focuses on new tools to define the genetic basis of neurological disorders. Very exciting session. The chairs are Robert Brown and Patrick Chinnery, and the symposia will describe current state-of-the-art approaches in neurogenetics, highlighting recent findings that influence our understanding of disease mechanisms. So the topics will be the role of genome-wide association studies, understanding and using next-generation whole exome and whole genome sequencing to diagnose single-gene disorders. We'll look at epigenetic mechanisms and RNA regulation. And finally, we'll look at mitochondrial disease and diagnosing mitochondrial DNA mutations. Attendees really, I think, will benefit from this a great deal. They'll have a much better understanding of our current tools and our current capabilities of understanding and caring for their patients. Our speakers are Stephen Saucer, Andrew Singleton, Mark Mailer, and Claes Wallstadt. So I hope you'll be here for that, be in Boston for that part of the meeting. In the afternoon, also very exciting, a look at modern methods for imaging brain structure and function. So this symposia will discuss ways in which the structure and function of neuronal networks can be explored using imaging tools. Now, a lot of research is centered on detecting pre-disease or molecular states, and that's quite useful. But in fact, we need to learn more about the pluses and the limitations of imaging methods. And so we'll have a good look at diffusion tensor imaging, fMRI. We'll really try to decode the bold signal and help us understand as neurologists exactly what we're looking at when we're looking at fMRI images. And we'll do molecular imaging studies as well. We'll look at that as a very useful way of thinking about brain structure and function. Our chairs are Kirk Fry and James Brewer, and our speakers are Michael Grachus, Lawrence Wald, and Joel Perlmutter. I hope you join us for this very interesting session, followed by the Special Interest Group Symposia. That's Sunday. See you there.